Oh, thank you, Declan. Proud grandfather. Text this morning, my youngest grandson choosing a pumpkin. It's almost as big as him. There we are. I loved Paul Wicks's opening start this morning. I'm going to be more emotional. Make sure I get the right button. You're asking me to do what? You're offering me treatment that doesn't work very well, and a computer will decide what I actually get, and all the treatments have serious side effects. Well, that was the reality I faced 23 years ago. And let's be honest, many chemotherapy clinical trials were, and some still are, similarly mad. Clinical research needs to think where the patient is coming from. Patients have a medical problem and they want to know what they've got, to know it's going to be treated, and to know they can be cured. Now remember, I'm talking about cancer here. It's all about having a clear direction and some certainty. Now, we know that certainty rarely exists. Clinical trials are about refining uncertainty. Therefore, for the patient, benefit versus burden sits at the top of the list of questions. Now, Derek Stewart planted that for me many years ago. I was treated on a palliative care trial in 2000. For me, it was about a cure, and it was a couple of years before I saw the contradiction. The process I entered only wanted my clinical data, and rather to their surprise, I survived. That was all that mattered to the investigators. Now, I understand the benefit-burden equation now in a different way. But the madness is complemented by magic because I actually survived. And humbly, I say I'm the only known survivor of that study. The last 4 CRFs in the study files in Brussels are all me. So, in 2002, I began to get involved as an involved patient. It started in fog, behind a net curtain, if you like, a mystery. It takes some time before you recognise that research is not an end in itself. So understanding how research conclusions get into clinical practice is something which patients too often ignore. We get bogged down in all the detail. We must learn about the big background stuff too. Trials should be there to serve patients. Thus, trials should answer the questions that matter to patients, not the questions the professionals think the patients want to ask, or that the professionals think that matter most. It can be quite different when you talk to the patients. There are some basics which the patients have to learn, of course. I had to learn the language to understand whether my view of the question I wanted to ask matched that of the respondent I was asking it to. And then I had to learn to understand the answers I was being given, as well as going on to consider the implications. The mist rarely clears, because in cancer research, toxic treatments are not unusual, and side effects are a key issue where patients want clarity. So how did I go about getting through the misty landscape to learn about methods? Neuroscientist Ian McGilchrist describes the way the human brain works following the route of science, reason, intuition, imagination. This is actually a systematic approach I like. But statistics? My first questions were about frequentist, get your finger around it, were about frequentist statistics. I did advanced maths at school, but it's not intuitive. Then out of the mist for me came the 18th century Reverend Thomas Bayes and his probability theory. I don't understand the maths here either, but the idea that no study is alone and that one should take account of the prior distribution of existing data when designing a study just seems logical. More like the science and reason leading to intuition described by McGilchrist. It seems much more like the way my brain worked. So back in 2007, I put together a patient-led session at the National Cancer Research Institute conference in the UK on probability and its potential. It was against the resistance of the UK science establishment, it must be said. Speakers included a researcher who was using a Bayesian approach in rare cancers. Today he is one of France's most influential oncologists, Professor Jean-Yves Blay. And today Bayesian statistics are accepted by all regulators, even if researchers are still reticent about using this approach. Then in 2009, I was involved with the HTA appraisal of a new drug to teach advanced soft tissue sarcoma. It was an innovative drug developed from a microscopic sea squirt from the mangrove swamps of the Caribbean. It had been in trials for some years, but because sarcomas are rare, there had only been small-scale phase 2 studies, although some had been randomized, but comparing dosage regimes. EMA licensed it as an orphan drug, and our HTA regulator, NICE, asked me to be the expert witness, as a sarcoma patient, on the appraisal. Now, there had been no quality of life research, so NICE chose an economic model based on advanced lung cancer and looked set to refuse use of the drug. Now, their model did not reflect the life that many patients have with advanced sarcoma, which tends to be active and functioning quite well until very close to dying, while lung cancer is a steady decline. So I challenged their choice of model, and I was supported by the specialist sarcoma clinicians attending the meeting. NICE threw out their experts model and remodeled based on our anecdotal evidence, and the drug was approved. Now, that experience confirmed for me that I needed to take a journey into what we called quality of life and patient-reported outcomes. But before I go into that in a bit more detail, I want to look at a few of the methodology issues which, as patients, we have learned about. Placebo. Comparators in randomized trials. This is an area of contention. As you know only too well, a scientific approach calls for randomization with a comparator treatment of known efficacy. Maybe a sugar pill, because nothing else is relevant. Can that be ethical? Patient pressure on placebo is huge and is having a growing influence. I know of Facebook pages where, through sharing experience, patients can work out whether they are on a placebo or not. Whether they then withdraw from the study or seek to get a treatment crossover if one is available, whatever they do, it'll defy the study design. I mentioned the crossover route, and that has its own issues. In 2010, NICE did not have a methodology for appraising trial crossover results, and the approval of one drug for sarcoma was held up for 9 months as a result. More madness. The question of comparators brought me towards pragmatic trials, where the comparator group are patients on standard of care treatment. I prefer this kind of approach. It gets rid of other biases in a study. Biases. Let's take a look at another two. Inclusion criteria. Brutally, as I see it, this is about how you create bias to give your study success. Anyone want to argue? I hear a chuckle, but no mutters. Right. Inclusion criteria are about deciding who is most likely to respond positively to the treatment. Although the argument usually used is to say that those who could be harmed are excluded. Surely you should have the whole cohort of affected patients included. Pragmatic trial design based on standard clinical practice and aiming to include all relevant patients, only excluding those who are known to have a high risk of an unfavourable outcome based on biomarkers perhaps. Surely this is a much better approach. Then, magic. Surrogate endpoints. A lot of theatrical magic is about creating illusions, and there are plenty of illusions in surrogate endpoints. PFS is the great abracadabra in cancer clinical trials. All of these, there's a little collection of 6 of them there, I think, 7 of them, are the great abracadabra. All of them are measures of how a drug has performed, not how the patients have responded. You can define PFS how you like. I'm not being sarcastic. It's true. A review of trial publications has identified 28 different definitions of PFS. Sometimes more than one is used in a single study. Some protocols do not standardize the way progression is measured. And who remembers to tell patients entering the study that PFS is not about survival, as it suggests, but about disease progression? That's very different. Amongst the other sources of bias, well, there's a little list of them there. Go on and on about them, frankly, but there we are. So let's look at patient-reported outcomes. PROs cannot solve all these problems, but getting to understand them and using them properly can certainly help. Find out how patients themselves report the effect of the treatment on their life and its quality. This is valuable data. Identify valid life contexts and question them. Pain, fatigue, gastrointestinal issues, they loom high in the patient's view. But there's also social functioning, physical activity, and cognitive issues, always important to identify. Techniques of measurement and analysis of PROs are continually developing. A recent addition to the work is identifying the MID, the minimally important difference, the point where benefit or burden actually comes into play. Now, I'm very keen on the electronic gathering of PRO data. And as you probably gather from my question to Paul this morning, the longitudinal gathering of data. And importantly, the issue of giving access to that data to clinicians so that when they meet patients in clinic, they have that data available to them and can discuss it with the patients. We are at a very early stage in getting that accepted as uniform practice. And the excuse is usually hospital IT systems. Well, I'm not going to go into that after Ronan's talk at the beginning of today. So let's just consider for a moment what's happening in cancer research now. There's a growing demand for rapid access to innovative medicines, which is challenging the regulators and researchers and the pharmaceutical industry. Precision medicine, innovative techniques such as CAR-T, new trial comparators based on retrospective data or using real-life patient cohorts, designs such as baskets, umbrellas, platforms and pathways. I'd love to know if there's more so I can extend that list. TWIC trials within cohorts, real-world data, additive to research data. I'm actually working on a study which is doing just that at the moment. Innovative statistics such as pairwise comparisons. Is that one you're familiar with? Shaking heads? A hand up over there? Pairwise comparisons, look it up. All this is happening, and all have methodology issues and deeper problems, the kind of detail that Kerry has just gone into. However, studies of treatment optimization are generally not happening. Where is the search for the minimum effective dose post-authorization, rather than blindly continuing to deliver the maximum tolerated dose, which was defined for the clinical trials? I know there's dose modification by clinicians locally, but that's not captured, and that's not fed back, and revisions for the authorization resulting. Where is structured phase 4 research focused on benefit and value for the patient, not just value for money? Not on the hot list for action as I can find out. It's got to be. As patients, we have to be involved in all these debates, and we must develop the influence which allows us to help initiate change. One problem is that those of us who are involved in this way are very few. Those holding the debates often work at a high level, unreachable through traditional patient involvement routes. Regulatory bodies are just not good enough at involving patients in anything other than superficial issues. Sadly, patient involvement is still mostly about ticking boxes. In this demand for rapid access to treatments, we are seeing a range of innovative trial designs, as I've suggested. And the recovery trial has been mentioned more than once. Run from Oxford University, it's looked at 12 drugs so far. A further 3 are currently being tested. Nearly 50,000 patients in almost 200 centres around the world have taken part. And even President Trump's hydroxychloroquine got looked at and comprehensively rejected. There's the TRACER-X study in non-small cell lung cancer, and the impact of that is going to be resounding for years. Its results culminated earlier this year in the cover of Nature, providing the article which gave inarguable data that air pollution is a cause of lung cancer in non-smokers. And that was just 2 weeks before London's extension of its clean air regulation for vehicles came into effect controversially. Now, neither Recovery nor TRACER-X could be regarded as a typical trial by historical standards. And both, I must say, have involved patients from very early on. So, a quick summary of what I'm challenging, I hope you will realise. I recall someone once saying to me, if we don't measure it, it doesn't exist. I now ask, what aren't you measuring, and why aren't you measuring it? If the analysis doesn't match the reality of patient life, we, as patients, must say so, loudly. I want patient-reported outcomes and experience measures sitting alongside traditional medical measures, analysed alongside them, and used clinically alongside them. Pragmatic trials with real-world cohorts are an important step forward. I recognise that surrogate outcomes can be important for eliminating confounding issues such as subsequent treatments, and can help speed patient access to innovative therapies. But extra rigour is needed when they're used, and there must be patient-reported outcomes, ideally as a co-primary endpoint. And interim licensing, particularly if a study has been accepted by the regulators on the basis of a PFS endpoint, that interim licensing must be accompanied by phase 4 studies run independently, not by the pharmaceutical company. Patients and patient groups can be innovative and challenging. Let's get the patient agenda around trial methodology articulated. Pharma is being diligent about getting patients to endorse its agenda. Regulators are trying to move beyond box-ticked involvement. Academic studies with groups such as you are getting really good at involving patients, and you're examining why and how you're doing it. So patience in the development of trial methodology is probably beyond its first stages and is becoming maturer and more considered. That's good. Well done. We, however, as patients, must start thinking beyond doing just the simple things. If you like, we must decide that we must not be manipulated. Thank you for your attention.